So this is part two of your Kin 425 lecture for January 27th. And this part involves the basics of glycolysis. So glycolysis involves the breakdown of glucose. It is a quick metabolic pathway, so it results in two ATPs being generated quickly, so a quick form of energy. And it generates two pyruvates, which can either enter the mitochondria, which can enter the mitochondria and produce even more ATP, or the pyruvate can go to lactate if you don't have that many mitochondria. So this is the basics of glycolysis. Glycolysis is this pathway where you have glucose going to glucose 6-phosphate. Eventually goes through a number of steps to get to pyruvate and then lactate. Pyruvate sometimes is referred to as pyruvic acid. The pyruvate is just the pyruvic acid without hydrogen, and same with the lactate. So the difference between lactate and lactic acid is lactic acid has hydrogen. The lactate is the ion that is separate from the hydrogen. So in physiological systems in the human, pyruvic acid and lactic acid dissociate into pyruvate and hydrogen, and then lactic acid dissociates to lactate and hydrogen. So here I've just written them as pyruvate and lactate. So some multiple choice questions to kind of lead you into glycolysis here is, which of the following enzymes is involved in glucose transport into a muscle fiber? The correct answer here is hexokinase. And the second question I have here is, which other enzyme is involved in glycolysis? And the correct answer here would be phosphofructokinase. So if we add those enzymes to this metabolic pathway, you can see the hexokinase up here. It's involved in the transport of glucose into the muscle fiber. The hexokinase adds a phosphate to the glucose, changes it to glucose 6-phosphate. The 6 just indicates where on the glucose molecule the phosphate is added. And that traps the glucose inside the muscle fiber. So this enzyme is very important for the transport of glucose into the muscle fiber. And then phosphofructokinase is just an enzyme that's kind of in the middle of glycolysis. And it's just easy to measure, so it's used as a marker of glycolysis in biochemistry labs. You have this other enzyme that's called glycogen phosphorylase, and that breaks off a glucose from glycogen, phosphorylates it, and changes it to glucose 6-phosphate. So glycogen is this carbohydrate storage in your muscle fiber that's made up of a lot of glucose molecules, and you phosphorylate them one at a time and change them into glucose 6-phosphate. The top part of this pathway is referred to as glycogenolysis. That's the breakdown of glycogen to glucose 6-phosphate. And then glycolysis refers to this bottom part of the pathway, glucose 6-phosphate down to pyruvate and lactate, and it includes this glucose coming in. So that's all part of glycolysis. And then you have this other enzyme called lactate dehydrogenase. And a key here is that dehydrogenase describes an enzyme that is involved in the exchange of hydrogen between one molecule and another. So in this pyruvate to lactate reaction, you're actually adding a hydrogen onto the pyruvate to change it into lactate. That hydrogen, it comes from NADH, which I haven't drawn in here, but that's where the hydrogen comes from. So the difference between pyruvate and lactate is that lactate just contains more hydrogen than the pyruvate. So important enzymes of glycolysis, you've got lactate dehydrogenase. It converts pyruvic acid to lactic acid. Or you can say it converts pyruvate to lactate. So remember, pyruvic acid is just the pyruvate plus hydrogen. The lactic acid is lactate plus hydrogen. Hexokinase phosphorylates glucose to trap it inside the muscle fiber. An important thing to remember here is that hexokinase is the only glycolytic enzyme that increases with endurance training. So that's one way by which endurance training could help to control your blood glucose levels. So having high blood glucose levels can lead to diabetes. That's one way that you can control diabetes with exercise training, is you get this increase in hexokinase inside the muscle fiber, and just allows more glucose to be transported from the blood to the inside of a muscle fiber. So it reduces your blood glucose levels. And again, high blood glucose levels is a risk factor for type 2 diabetes. So a question here is, what are two possible fates of pyruvate during exercise? And then what enzymes are involved in these reactions? So from what I've covered, you should know one possible fate is that pyruvate can go to lactate. So that's what I have down here. And that's catalyzed by that lactate dehydrogenase. So that lactate dehydrogenase adds hydrogen to the pyruvate to change it to lactate. The other possible fate of pyruvate is that it can go into the mitochondria, and that involves a mitochondrial enzyme called pyruvate dehydrogenase. So that's what the PDH stands for. You can see that here the pyruvate is a molecule that's made up of 3 carbons. By the time it goes into the mitochondria and is converted to this other molecule called acetyl-CoA, there's only 2 carbons left here. So coming off of this reaction, you have NADH, and I wanted you to kind of fill in the question marks here on what is released during this reaction where you lose carbon molecules. And the correct answer here is it is carbon dioxide. So this carbon dioxide is part of the carbon dioxide that you would breathe off during exercise. So during exercise, you have activation of this metabolic pathway. If pyruvate goes into the mitochondria, it gives off this carbon dioxide, and you get an increase in the carbon dioxide that you actually breathe off from your lungs, and that leaves your lungs and goes into the atmosphere. So two fates of pyruvate, you get formation of lactate, or you get pyruvate entering the mitochondria and being converted to this acetyl-CoA. And in that process, you have release of carbon dioxide, and you can also see you have production of this NADH. And I'll get into the importance of that NADH a little bit further into this lecture. So the bottom line here is that if you have very few mitochondria, it's more likely that your pyruvate is going to form lactate. So that's one characteristic of someone that's untrained, is that at a given exercise intensity, they will have a high production of lactate or lactic acid because they have few mitochondria. The pyruvate is being driven to produce more lactate, and there's not a lot of pyruvate that's being taken up by the mitochondria. Whereas if you have a lot of mitochondria, it's more likely that your pyruvate is going to make it into the mitochondria, go through that pyruvate dehydrogenase reaction, and enter into aerobic metabolism. Now, you also have this enzyme, an isoform of lactate dehydrogenase that's in the mitochondria called lactate dehydrogenase H. Lactate dehydrogenase also converts pyruvate to lactate, but this isoform of the enzyme is a little bit different. You can see it's called the M form, whereas the one associated with the mitochondria is the H form. So the M form converts pyruvate to lactate. The lactate dehydrogenase H form converts lactate back to pyruvate. This M stands for muscle. This H stands for heart. So this would be very high in cardiac tissue or heart tissue because your heart is like a very aerobic muscle. So this is just a summary of the lactate dehydrogenase isoforms. You have the M form stands for muscle, converts pyruvate to lactate, and then you've got the H form that stands for heart, converts the reverse reaction, and this occurs in the mitochondria. This lactate dehydrogenase M reaction occurs in the cytoplasm outside the mitochondria. So this is just another thing that can happen with endurance training. You have a lot of lactic acid being formed if you're untrained. And then after training, if you have a greater number of mitochondria and a greater size of the mitochondria within the muscle fiber, and that's an adaptation with endurance training, then you can get greater lactate being taken up by the mitochondria through that lactate dehydrogenase H reaction. So this is kind of a summary, the effects of aerobic training. You get an increase in the number and size of mitochondria in your muscle fibers. It increases the chance that pyruvate or lactate formed during glycolysis can be taken up by the mitochondria and be oxidized. It decreases the buildup of lactic acid. So that's a characteristic of an endurance trained person. So a very highly trained, endurance trained person can exercise at a very high work rate, so a very high intensity, without having that great of a buildup of lactic acid. And that's because they have a lot of mitochondria in their muscle fibers, and the mitochondria are going to take up pyruvate before lactate is formed, or if lactic acid is formed within the muscle fiber, the mitochondria can take that up before it appears in the blood. So if you're taking blood measurements from an endurance trained athlete during an exercise test, often it's very difficult or it's very rare that you see a high level of lactic acid being produced. And this leads to a decrease in fatigue because lactic acid interferes with a lot of the steps in muscle contraction and causes fatigue.